So I will tell you about the PacMAN project. PacMAN stands for Pacific Islands Marine Bioinvasions Alert Network. Now, what this project wants to do is build a capacity in ocean science and observations, especially for early detection and rapid response to marine bioinvasions in the Pacific Islands. This project is managed by our group at the OBIS, so Ocean Biodiversity Information System, as well as our collaborating management group at the University of South Pacific in Suva, Fiji. I'm going to tell you shortly about the background, so the problem that we are trying to address, the structure of the project and the focal points that we have, shortly about the methods that we're going to use, and a bit more about the data management workflow, as well as the decision support tool that we plan to develop. So the problem here is invasive alien species. These are considered to be perhaps one of the greatest threats to marine biodiversity. These species move with ships' ballast water and vessel biofouling, so through human activity and marine traffic. Now, marine traffic is constantly on the increasing, so also the risks posed by these species are increasing. These bioinvasions pose a real biosecurity risk for human health and the sustainability of livelihoods, especially in small island developing states, which are hugely dependent on the marine environment around them. In addition, eradicating already established species is extremely difficult, so it is very important to be able to identify the risks early of newly arriving species in the environment. Now, this is where PacMAN is trying to come in. So what we want to do is enable a monitoring system which would allow these Pacific Island states to quickly detect newly arriving invasive species in their environment. We will be working closely with partners and stakeholders in Fiji. So we will be identifying the needs that the local community has in terms of invasive marine species, as well as aligning our project to already existing initiatives in this area. We want to be developing the monitoring plan with the use of environmental DNA. And this is because environmental DNA analyses can be accessible as well as rapid. They can be, you don't need a huge amount of expertise to be able to do DNA analysis and sampling in the environment. We also want to be developing standardized bioinformatics and data management workflows that would enable an easy way to analyze the data coming out from environmental DNA. And this would allow this project and these protocols to hopefully be disseminated to other areas that might need similar monitoring programs. Ultimately, we want to develop a decision support tool for the early warnings of invasive species presence that can be utilized by the environmental managers making decisions in the area. Because we want this project to be something that can work on the long term and can stay in the area even after this project term is over, we will be having a training program for local scientists and environmental managers for the protocols developed in this project. Now, very shortly about the methods. So we will be using environmental DNA and metabarcoding to detect marine invasive species. We will develop protocols for sampling, sample processing, and molecular analysis for organisms and biodiversity, especially in high-risk areas like ports. We also want to do an initial baseline survey to look into the biodiversity here, as well as what could be considered as invasive. So we want to have a long-term program that then can look into the previous samples as well as the control samples to see which organisms might be actual new invasions in the area. In addition, we want to make sure that through these methods, we are reliably identifying the species, so annotating the sequences to the right taxa, as well as classifying these taxa in a right way to either invasive or indigenous species. However, a big part of this project is the data management workflow. And PacMAN will in fact be one of the first times that practical work on environmental DNA analyses will be directly linked to the OBIS database, and therefore will act as a pilot for the development of a standardized pipeline for genetic data addition to OBIS. We have a preliminary plan for how our workflow could look like. So this is the data and metadata processing workflow from raw sequence reads to standardized OBIS occurrence data. Here, if we start at number 1, so what we want to do is already link our metadata to the raw sequence data that we get from the sequencers. This raw sequence data will then go through a bioinformatics pipeline to be analyzed into informative sequences, where it will be pre-processed, denoised, and annotated. The annotations will be done against reliable reference databases that are curated and updated regularly. We will get out the amplicon sequence variants, which we hope to deposit to an ASV repository. This is something I think that is currently being discussed, and we are very much interested in joining efforts and joining these initiatives. Then the ASV tables and taxonomic tables will be ingested into the biome format, which will be then combined with the original metadata into a package that can be ingested by OBIS and transformed into the standardized Darwin core formatted data. And then this data will inform the decision support tool. So we envision this decision support tool to become an easy-to-use dashboard that will provide the environmental managers with early warnings of invasive species presence. So how we see this to be, so this decision support tool will use habitat suitability models for the detected species. It will make risk assessments of any of the non-indigenous species to see the likelihood that they could establish themselves in the new environment and become invasive. And it will provide alerts on these detected species. So how it will work in practice is we will use the species occurrence data from OBIS and the ones that we detected, as well as environmental data from other global databases to model the habitat suitability of these specific species, as well as compare this to taxonomic databases of invasive species and their traits. This will assess the likelihood that this species that has been detected could establish itself in the new environment based on the environmental parameters, and it will provide a warning for any species that could pose a risk or are known to be invasive in other areas. These risks then will induce a full risk analysis from the environmental managers to see if and how much action should be taken in terms of these newly arrived species. So as you can see, we are in the beginning stages of this project and in thinking about all the details that go into it. And we are especially interested in having discussions on how we will align adding genetic data to OBIS with other initiatives that are going around in the community. So we want to discuss how exactly sequence data and metadata will be linked so that it will be according to the Darwin Core archive standards, but also compatible with other standards like the MixS. And our goal ultimately is to have reliable and comparable occurrence data. Now for sequence data, this means that we would want to have an ability to harmonize and update the taxonomic annotations of the sequences regularly, because we know that these reference databases are constantly being developed and updated. We also want to be able to check distributions of taxa that are currently dark, so that are not yet annotated reliably to any scientific names, because we know that these sequences form a big part of the sequences detected in the environment. So we don't want to lose this data at this point. So that was my short introduction to PacMAN. As I said, we are open to recommendations and discussions. We are also in the working group of TADWIC for genomic biodiversity, as well as the task group on Darwin Core and MixS alignment. In addition, we have a project team for developing guidelines for the addition of genetic data to OBIS that will be active in 2021. So this is open for any people who would like to join the discussion with OBIS. So don't hesitate to contact us in terms of that. Thank you very much.